recap about you, Hannah.
Okay. So
Yeah.
should I do the recap, sir?
Yes. So let's start with immunity.
Okay.
What
Uh,
we did. What have we done till now?
Immunity is like the overall ability for the organism to resist or fight against the disease-causing microorganism is called immunity. There are two types of immunity. One is innate immunity and adaptive immunity. Innate immunity is like an inbuilt or like by birth a general type of immunity. Adaptive is when a person adapts to that particular how to say disease or cold, I guess. So it immunity increases by that, and then innate
Immunity increases by what?
immunity is a non-specific type of defense.
You were saying something about the immunity, adaptive immunity. It increases by that or something like that. I missed. I think your voice was
Yes.
not audible. Yeah.
Adaptive immunity is like when a disease attacks you the first time, the
Okay.
organisms would know next time, like the second time when an organism comes again, that same disease comes again, the how to say,
Body will know. Your
yeah,
immune system will know.
immune system would know how to attack, where to attack. So that's the thing.
Correct. Correct.
And innate immunity is non-specific type of defense. So there are four barriers or layers. Okay. The first one is physical barriers like skin. Okay, skin is dead. It can't. We cannot. Nothing can affect it. Okay, and then mucus. These are
It's
the
not like nothing can affect it. It's not all dead. But the first layer, few layers, like the top layers are not living cells. So that is what works in our favor, right?
okay, so
The skin also has living component. Yep.
one minute.
Yeah, sure.
Yeah. Then is physiological barriers, which is acid in the stomach and saliva in the mouth, and tears in the eyes. These are the few ones which we discussed, few examples. And then cellular barriers are leukocytes, which are WBCs, and then neutrophils, basophils, um, monocytes, and lymphocytes. Lymphocytes are natural killers. These are the like,
One type of lymphocytes are NKCs, natural killer cells,
yeah, oh yeah, yeah, yeah.
because other types are the B and the T lymphocytes, which I will be teaching you today.
Okay.
Yeah. So the type of lymphocytes that contribute to the innate immunity are NKCs, natural killer cells.
Yes, sir.
Yeah.
And
Continue.
then we went for macrophages in tissues are phagocytosis of microbes.
Phagocytosis.
Yeah,
Phagocytosis is eating the microbes or
yeah.
degrading the microbes. Yeah.
Then the last one is cytokine barriers. Here is the place where, like, um, proteins. But no, sorry, my bad.
So cytokine barriers are basically molecular.
Yeah.
Yeah. So interferons are the players for cytokine barriers. And these are protein molecules which are secreted by the virus infected cells.
Okay.
So those cells which are infected by a virus and are most likely to die themselves, they secrete these interferons, which helps the adjacent cells or the nearby cells which are non-infected to protect themselves and to stop the spread of infection.
Okay, so.
Yeah.
And then there's acquired immunity. It is a pathogen specific. It has memory. Then you are like memory. What does memory mean? When a body encounters a pathogen for the first time, it produces response known as first encounter as a primary response. Then it is low intensity and a slow response. And then when the second encounter comes, it's faster and high intense response because it knows where to attack and how to attack.
Correct.
Then it's carried out by two special lymphocytes. One is it's called as BT lymphocytes, I guess.
It's called B lymphocytes and T lymphocytes.
Yeah, okay. B lymphocytes and T lymphocytes. The B is bone marrow and then T is thymus.
So these lymphocytes,
yeah,
these lymphocytes come from these two
yeah,
places.
and lymphocytes produces B lymphocytes produces antibodies, while T lymphocytes help in the B lymphocytes to produce antibodies.
Yes. Correct. So I think this is where we stopped in the last class, and I told you that in the next class we'll be doing in detail about the antibodies and everything, right? Okay. So we have started acquired immunity, and you realized the broader theme of acquired immunity is that it's pathogen specific and it has a memory, correct? Now, also, the first encounter with a pathogen, which is known as the primary response, you have to remember that it is mostly low intensity and a slower response as compared to the second encounter with the same pathogen. That's called secondary response. So secondary response is very intense and faster, and it's also known as anamnestic response. Now, both first encounter and second encounter is carried out by these lymphocytes. So these two types of lymphocytes, B lymphocyte and T lymphocytes, they're named B and T because of where are they synthesized and produced in the body. B lymphocytes originate and come from bone marrow majorly. T lymphocytes come from the thymus, hence the name B and T lymphocytes. Okay. Now, as I told you, B lymphocytes produce antibodies. So today we'll start with the structure of antibodies. So write down. What is the date today? It's 28th, right? Okay. So antibodies. Anyone has any idea what antibodies are? No one has no idea. You must have heard the word antibodies, antibiotics. Any idea what it is? Where have you heard it in your real life, apart from the textbooks? Never heard of antibodies? I'm sure you must have.
Vaccines. Vaccines are antibodies. I think.
Sorry, your voice is very muffled.
Vaccines. That's
Vaccines.
right. Yes. They're
Where in
antibodies.
vaccines have you? So my own voice is getting echoed through your. Are you on speaker or something?
No.
It's Bluetooth or earphones. Probably. Okay. So that might be having some issues. So you said vaccines. So what in vaccines? Where have you heard the word antibodies in vaccines? And I am suppose that you're talking about the COVID vaccine, right?
Yes. They contain antibodies.
Do they?
Yes. Because antibodies are the ones which are sent to like gain immunity in your body. So when the actual disease comes, affects you, it can fight back because it's already used in your body. Like it's already used.
Yeah, but who told you that the vaccines contain antibodies? You must have heard it or read it somewhere, right? You didn't think of it on your own. But where did you read it, or from whom you heard it?
From Google.
From?
Google and like vaccines, like how vaccines work, and when I was into that.
Okay. Yeah, so how vaccine works makes sense, right? But we'll come to this part, whether I'll be talking about COVID vaccines a bit, in some bit. But so what do you know about, you already told me that antibodies are certain things which helps the body fight against the pathogens, right? How? What do the antibodies do? Where do they come from? How do they know that they have to help the body? How are they formed? Why are they formed? Are they pre-formed? So do we have antibodies right from the birth in our body? Do we have antibodies against pathogen from birth?
Yeah, so we do have antibodies right from birth.
Against what?
Against let's say survival, I guess.
Against?
Like there are so many pathogens around the area. And
Exactly. So how do you think those pathogens reached to you when you were in the womb? How does your body came to encounter them so that you have it right from the birth? To have it right from the birth, the pathogen must have come and encountered, right? That's where antibodies are formed.
now, suppose if the mother catches a cold, like the baby's antibodies recognizes it and tries to prevent that particular pathogen, I guess.
Yeah, you're very close, but baby does not have any antibodies right from at the time of birth. The baby only has innate immunity. That's what I taught you. The only thing which we have by birth is innate immunity, and innate immunity does not consist of antibodies. What I was trying to push you towards an answer. You're right that sometimes babies are born, or when they're born, they already have some antibodies in their body, but that's given by mother. So instead of saying that mother's pathogens are fought by the baby's antibodies, mother passes some antibodies through the placenta to the baby. So those are not built by baby. Okay, those are not produced by us. Also, after the birth, the colostrum contains antibodies, the mother's milk. So the baby should be fed with the mother's milk properly, and for an appropriate amount of time for proper development and also the immune development. So babies can only form antibodies after they are born, after they encounter real pathogens in the real world themselves. Before that, if they have it in their body, it's coming from the mother. They cannot build it when they are in the womb because they have no exposure. Do you understand?
Yes, sir.
Now there are, it's not like biology does not talk in discretes that this will happen or this will not happen, black and white. There can be certain kind of infections that can also cross placenta. For that, the baby's immune system, when it is even not developed, tries to produce some antibodies, the B lymphocytes and the T lymphocytes. So that way also, but those we are not going into the detail of because that's a complexity. So I want to keep the demarcation of innate versus acquired separate by the time of birth. Okay, it's not like the baby can never make antibodies. It can make antibodies, but normally it happens after birth. Before that, mother gives whatever is needed. Make sense to you, Hannah?
Yes, sir.
But what are antibodies, if I ask you? So antibodies are basically nothing but proteins. Okay, they are proteins, and there are so many numbers that it becomes an army of the protein. So our cell, our immune system creates an army of proteins that can go and fight with the pathogens when they enter our blood. Do you understand? For antibodies to work, the pathogen has to be in the blood. Okay.
Yes, sir.
So write down. An antibody is a multimeric protein. What do I mean by multimeric protein?
It has multiple varieties of protein.
It has multiple components that assemble together to become one antibody. Okay.
Oh.
So very specifically 4. So it's also known as, so write down, I'll just dictate it to you. Antibodies are multimeric proteins produced by B lymphocytes. To fight against the pathogens. So they are proteinaceous. Okay, we got to know that antibodies are proteinaceous in nature, so they are proteins. Now write down, each antibody molecule has 4 chains. Okay. So 2 are bigger chains or heavier chains. We call them heavy chains. And 2 which are depicted in green are light chains. And they are connected together by a sulfur linkage. Okay, SS linkage. Sulfur. So this heavy chain and this is a light chain. So this structure is known as H2L2, heavy 2, light 2. Make sense? Yeah. And this side is the N terminal and this is the C terminal. In every protein, there is a, just like remember about DNA, there is a 5' end and there is a 3' end. Remember? Yes, no?
Yes, sir.
Yeah, so for proteins, there is a C terminal and an N terminal. So this sulfide linkage joins every part of the antibody. So the long chain with the long chain, the long chain with the short chain, the long chain, the short chain with the short chain itself. So all these linkages are sulfur linkages. Is it clear? But overall, the structures remains like this. So this is called disulfide linkage. Okay, disulfide linkage. Now there are some regions that you should know. So this region, I'm marking it with this region here in an antibody is called antigen binding site. And how many antigen binding site does one antibody has? How many? Antigen 2. So each antibody can bind to how many pathogens?
2.
2. So let's say this is a pathogen. Okay. So the antibody, sorry. So the antibody will bind like this, right? With one pathogen. This is the Y-shaped structure. I'm not making the light chain. Let me make the light chain also. Correct? And on this side, it will bind to the second pathogen. Make sense?
Yes, sir.
Similarly, because this pathogen can still, there is another antibody can just bind here. Make sense? Because this antibody has just bound to one pathogen, so it can now here bind to another pathogen. Do you understand everyone what I'm trying to do here?
Yes.
Yes, sir.
And for this same antibody, there can be another pathogen, sorry, another antibody which can bind to here, and then let's say it binds to one pathogen here. So what's happening basically? Can you tell me how is antibody, what is antibody doing basically? Antibody antigen reaction or antibody pathogen reaction, what is it like? Antibody, so these pathogens were independent, let's say they were bacteria, so these were independent cell. This wanted to go somewhere and infect the cell, this wanted to go somewhere and infect, this wanted to go somewhere and infect the body. But what happened? Ultimately antibodies made a cluster of all these, right? They coagulated, agglutination it's called. So now the pathogens cannot function, cannot go, like it's like they are trapped in a mesh of antibodies. You understand?
Yes, sir.
So antibody antigen reaction always mostly is agglutination reaction, where something just precipitates or forms the bulk or the clot kind of thing. That's why every antibody has two antigen binding site, which it can bind with which it can bind separately to two pathogens or antigens. Make sense? If it's a living substance like bacteria, we call it pathogen. If it is a chemical, that's toxin or something secreted by the bacteria, it's called antigen. Okay. Now in our body, there are different types of antibodies. Anyone knows? Human, what are the different types of antibodies in human immune system? So one is IgM. Now this Ig, the word Ig, you know what does the word Ig stands for? It stands for immunoglobulin. The word immuno means immune system.
Immunity.
Yeah, immunity, immune system. And the word globulin means protein. Okay, simple. So it is protein that helps in the immune system. So it's immunoglobulin. Now IgM, M tells about what kind of antibody it is. So there are antibodies IgM, IgE, IgG, and IgA. Immunoglobulin M, immunoglobulin E, immunoglobulin G, and immunoglobulin A. Now I remember them through this word called mega. So all the mega response in our body to fight against diseases, to keep us healthy, comes from mega. You can also remember it as game. So these antibodies play a game with the antigen. That's the game of defense and attack. Is it clear? MEGA.
Yes, sir.
Out of this, easy, I'm just giving you some shorthand tips too. IgA is very, very, like it's small in size, and IgA can transfer to babies from the mother. Okay. And how can this transfer happen?
Through the placenta?
Yes, through placenta and colostrum. You know what is colostrum?
Yes.
Yeah, what is it?
Yes, sir. It is like a yellowish fluid. Fed. No, it's like a yellowish fluid that is like produced in the mother's breasts during breastfeeding. It contains a lot of
Yes.
antibodies.
Yeah, you could just say it human milk. Human secreted from female human's mammary gland is colostrum. Okay, so mothers towards the end of the pregnancy, they come into the period of lactation. Just after parturition, lactation starts and they start producing milk in their mammary glands, right? So the milk of the human is called colostrum. And as Atif said correctly, it's thick and yellowish. Okay, and it has antibodies, especially and IgA is there a lot. Do you know whenever there is an infection or inflammation in the body or allergy in your body, which antibodies reaches the site of infection first? Any idea?
No, sir.
Okay, remember that it's IgE. Write down IgE. IgE reaches the site of an inflammation or allergy, or are produced during allergy. You can write like that. IgE are the antibodies which are produced during allergy. IgM is very big. It is the biggest of these four, and it is pentameric antibody. What does it mean?
It has five parts.
Yes, it's multimeric antibody. So these are just few things you should know in general about the human antibodies. There are 4 types, etc., etc. Now, as I told you, antibodies are produced in the blood, and we'll come in a bit to COVID and try to understand where Fatima has heard about the antibody in that context. But you understand that these antibodies are always floating around in our blood. So whatever infections we have gone through till now in our life, it's possibility that we have antibodies against them stored as a memory in our body. That's why next time when there will be the same pathogen attacking, we can mount a better response. Is it clear, everyone? Yes. So because these antibodies are present in the blood, write down immunity through antibodies immunity through antibodies in the blood, because it happens in the blood, is known as humoral immunity for blood. Humoral means blood driven. Okay. Now this is one type of acquired immunity which works through antibodies. And which cell makes antibodies? Secreted by B lymphocytes. Is it clear?
Yes.
So B lymphocyte driven immunity is called humoral immunity, antibody driven immunity. Okay. Now apart from that, what happened to the T lymphocyte? What do they do? What do T lymphocytes do, according to you? Yes, they help the B lymphocytes produce antibodies, but just that? So T lymphocytes mediate another type of, so this is the first type of immunity, right? First type of adaptive immunity. Now let's go to the second type. So for that, we have to go to T lymphocyte driven. T lymphocytes immunity. Okay. So write down the T lymphocytes mediate, or the T lymphocytes are important for, write down T lymphocytes are important for the cell mediated immunity. That was humoral immunity. This is called cell mediated, that is blood mediated. Cell mediated immunity. इसके कुछ features we'll read about it. Anyone knows what is a cell mediated immunity? It's mainly driven by T lymphocytes. So anyone has any idea? I asked you to read about a little bit on immunity, right? You did not read. Cell mediated immunity. How many of you know about organ transplants? Organ grafts or organ transplants? No one has heard? Come on, people, participate. If you're not speaking, then what's the point? It's for you, right? I know all these things. So you have to participate. What happens during a organ transplant? What is an organ transplant or a tissue transplant? Or we call this technique as grafting. Where else have you heard this word grafting? Let me give you a hint. Plant system. Am I audible? Hello? Or not? Hello? Am I audible?
Yes, sir.
Yes. Okay. Transplants. Never heard of the word organ transplant, tissue transplant? No?
Yes, sir.
What is it? What is a transplant?
So, like, if a person's organ is failing, they
Mhm.
I guess one of
Yeah, you don't have to guess. I think it's as normal as every fifth
Yeah.
class student knows what is an organ transplant.
and then they
So we have a donor, we have a recipient. We take a donor's organ which is in the good working condition, and we replace a recipient's organ which has failed or which is not working properly. So that's like replacing hard wires or replacing hardware of our body. Yeah. But is it easy? Is it very doable?
No, sir, it's very hard.
What's the problem? Why is it hard?
Sir, like first we should check whether the person is the same blood type.
Okay, why are we checking that?
Because like if it is a foreign blood type, then the body might mistaken it as a foreign um, how to say material, and then it will start attacking it.
So why doesn't it attack the blood? We all must have donated or received blood in our life. Blood transfusion is a very common thing, right?
Yeah.
People donate blood, so that's not that's also foreign particle. Any cell from my body is going to be foreign for your body. So what are we checking for? And why some foreign particles getting accepted by the body easily, and some foreign particles have all these nakra, they don't get accepted, there'll be a reaction or there'll be attack. What's happening here? See, you have to think, right? Have you never thought of that every blood transfusion is actually a foreign particle coming in someone's body, but it saves life?
But aren't they the same blood type?
How can, but you said that foreign particle, the foreign body. So blood is coming from my body. To your body, it is always going to be a foreign particle, right?
Yeah.
Yeah. So will your body attack that blood? So you used another word called same blood type. Yeah. So what does that mean? Let's first define that. What do you think by same blood type? Yeah.
Same blood type in the sense. Now, suppose if I'm A positive, I should.
What is A and what is positive?
God.
Yeah, of course. See, this is what they are going to ask you in your entrances. We have just memorized things in our life. We know nothing about what's happening around us. If I ask you, there's too much of sun, so you'll say, okay, use sunglasses. Now if I ask you, how do sunglasses work? Oh, oops, don't ask me that. Let's put sunglasses and go out. We all use mobile phones, right? You know, why doesn't mobile phone works in aeroplanes? But now it will work. Now from 5G onwards, you know, even aeroplanes, you can use mobile phones. So what's happening there? How is it working? No, oops, we don't care about that. So we use science, but we don't care about science. So the only option left is to just memorize some facts that blood transfusion can be done. Just match A positive with A positive. But what is A and what is positive?
It is like a category of blood.
So what is that category? How have we decided? On the basis of color? Oh, this looks like very red, so it is.
No, not color. Like I think it's shape.
It's associated with what I'm teaching you actually. Yeah. What is there on basis of which? Yeah.
I think it's shape.
Shape of blood.
I don't know. I'm not sure.
Can blood have a shape?
Yeah, that's like a
Blood is
shape.
blood is a liquid. How can it have a shape?
That RBC cells. Right?
All RBCs have one shape only, I think. In human species specifically, all our RBCs are biconcave in shape, discoid. We don't have different shaped RBCs. In fact, when the shape of RBC changes in sickle cell anemia, we get a disease. You all know this. Chapter 6 I taught you, but the problem is no one ever goes back and thinks that today I have learned something interesting. Let me share it with my parents or a colleague or a friend, or let me share it with myself. Let me sit with that knowledge in the class. So you are still studying for examinations. Don't worry about that. All of you will get great marks in biology, that I can ensure you. I can give you in writing, because we cover more than what you need. You already need certain facts. You have memorized certain facts. For that matter, there are 75 questions that you can remember and go for boards. You will pass in the boards with surety, guarantee. That's a guarantee that tuition centers and coaching centers take. But that guarantees for those who are miserable students, those students who just want to get 35% marks or 40% marks and pass that subject. So if there is a guarantee, so there is a guarantee to pass the subject. There is never a guarantee that you will understand the subject. For that matter, whatever subject it is, unless and until you want to understand it. So it's biology is everywhere around you. So blood is categorized, and others, those who are not at all speaking, are just wasting their money, And I'm very, very brutally honest about it. I think the beauty of this system, why I teach on this platform and not on other platforms, is that I don't want a class with 50 students. It's not like I have not been approached for that kind of things. Yeah. And even, 50 students in a class, everyone's mic is shut down. Even my camera, I try to keep my camera on as much as I can. And I will appreciate one day. There are few students who deliberately keep their camera on, and it feels like I'm with a student in a discussion. But anyone who wants to keep their camera on, please do that. Because I think you should tell your family and parents that see, this is my studying time, I'm going to a class. This option was not there to hide behind a curtain if you were going to an offline class paying the same or even more amount. Will you agree with me? You cannot be invisible in an offline class, right?
Yes, sir.
Computers have given you this privilege, which I think works against the students. I hate teaching in an online mode. I still do, If you give me option any day to teach between online and offline, I'll choose offline without any bias. Oh, sorry, with the bias. I am very much biased because you are there. You committed to come and be there physically. And now if you are not mentally there, then I can just pin it down on you. You are here, you should better focus. But on online mode, I don't know many of you, what are you doing when I'm just speaking? When I call your names, you come back. So that's always there. But I'm telling you, if you are investing in Learnivio, the model is that we keep the classes size small. You know how many of you are in this class? Five people with me. But 3 out of 5 will speak nothing throughout the class. Then you better be in a class where there are 50 students. There's no positive benefit you are taking from this model or from me. So that's what I'm trying to tell you. That's all. It's up to you. I just, if I don't, when I don't enjoy teaching, then I just teach because it's my work. But when I enjoy teaching, that's a good thing, right? Everyone should enjoy what they do. Yes. So blood group are actually categorized on the basis of antigens present on their surface. They're just studying antigen antibody, right? So A and B are two type of antigens which are present on the surface of the RBC. So if I have only the gene for antigen A from both the parents, which means my blood group is A, okay, and positive means Rh factor. It is again an antigen, Rh. So A, B, and Rh, these are the three antigens which are the major antigens present on the RBC. Now it is very important to see that the blood that we are donating to the person, their blood group should match, because if I put a blood group in a B blood group's body, remember that if my blood group is A, so antigen A is present in my body. Yes or no?
Yes, sir.
So I will not make antibodies against my own antigen. Otherwise, I will start killing my own RBCs. Yes. So I have no antibodies against antigen A, but I will have antibodies against antigen B because that is not my. Do you understand? Or if I do
Yes, sir.
not have, when you will put that blood in me, I will not accept it as my own blood because the antigen is different and I cannot recognize the antigen. So I will make antibodies and try to destroy that antigen. My body will try to destroy antigen B. So a B blood group in a A blood group's body will cause a immune reaction, a big reaction that can give shock to the organs or even kill the person. Do you understand now? Why do we need to match blood before transplantation or something?
Yes, sir.
Yes, sir.
Simple, right? Logic. So A, B, and what is blood group O? O is not,
No
O
antigen.
means, yes, O means 0, nil, no antigen. So neither A nor B. Now the problem with O people is, O negative are called universal donors. Why are they called universal donors?
They
think.
can donate to any blood group. To people's any
Why?
blood.
Why they can donate to any blood group?
They do not have any antigen.
Exactly. Neither they have A, neither they have B, neither they have Rh. O negative. So you can just take O negative blood, and if it's an emergency, you can just transfuse it into anyone's because they don't have any antigen. So the person who's receiving that blood will not start a reaction against the blood because no antigen is there. Make sense?
Yes,
Now
sir.
you tell me, which blood group will be universal recipient then?
I think it's
If
positive.
O positive,
Yeah.
you're not thinking through.
AB positive.
You are also not thinking through. Universal recipient.
Receiver, right?
Yeah. Sorry, yes. Did you say AB positive? Anyone?
Yeah.
Yes, sir. I said
Yeah, he said.
Yes, yes. So why will AB positive be universal recipient?
because they have every antigen in their body.
So that's not the answer. If they have all the antigens in their own blood, what they will not have?
Antibody.
Exactly. So their blood will not have any antibody. So whatever blood you put in their body, actually it will not cause reaction to that blood. You understand? AB positive. Whether I put O positive, O negative, A positive, A negative, B positive, B negative, AB positive, AB negative, no, all the antigens are accepted in AB positive's body because there is no antibody. Make sense or not?
Yes, sir.
Very good. So again, this is the same concept. So for transplantation, we need to match certain things. Like Hannah very correctly said that we have to match the blood group. We understood the blood group, but just blood group matching will work. Suppose I want to do a liver transplant and I match the blood group. It matches. Hello, am I audible? Sorry, I lost the internet connection at my place. Am I audible to everyone?
Yes, sir.
Yes.
Yeah. So just before I lost the connection, I was saying that suppose the blood group matches, okay, Hannah? So that's it. That's the green signal. I can put anyone's liver in anyone's body. Think about it. So if the blood group matches, many blood groups, so blood group can match very easily, right? There are only 4 types of blood groups. Yes, A, B, AB, and O. 8 types if you consider negative and positive different, let's say. There are 8 types of blood group and there are more than 7 billion people. So multiple people will have same blood group. So which means then why is organ transplant such a big deal? We can transfuse blood every now and then. You know, we can donate our blood. It can go to someone very easily if the blood group matches and if your blood does not have any infection or pathogens. Simple. But then why is the organ a problem? Clearly, it's not just blood, right? we also have to match the organ matching, the tissue type, the cell type also should match. So every organ has a characteristic property, a chemical property, biochemical property. So that should also match the receptors on organs. So any tissue that comes from outside in the body is a foreign particle, including blood. So I just told you, am I audible to everyone? Because I'm going to tell you something interesting here.
Yes.
Yes, sir.
Yeah. So I told you that if the blood group matches, so problem is solved. It's not like that. There is always a reaction, always, because A, B, and RH are not the only three antigens which are present on the surface of RBC. There are more than 200 antigens that are present on the surface of RBC, but what matters basically, which are the most abundant and causes the most immunogenic response, are these. So they are the major. So ABO blood group is the most accepted blood grouping system. But when you go for organ donations or organ transplantation, they check more than that. Even in the blood only, they check more than just A, B, and O. And oftentimes, when they take organ from a person, they don't take that person's blood. So the organ, whichever organ is to be transplanted, first all the blood is taken out from that organ. So it's kept in a solution where all the blood leaves the organ. And then you put that organ in the person's body and new blood starts filling each and every artery and capillary or whatever in that organ. Okay. So, but the organ also has to match. So there is a tissue matching also, that's important. And that's even more complex. So that's why finding a donor that matches you is very, very difficult in the first place. Even if you find it, that will not be 100% match. There can never be 100% match unless and until you have an identical monozygotic twin, because the genetics will be same then. You understand?
Yes, sir.
Identical monozygotic twins have everything same, the blood group, the antigens, even the tissue type, cell type, to a major extent. Do I make sense?
Yes.
Yeah. So even in blood transfusions, there is always a reaction when someone's blood enters your body. Even if it is the same blood type, there is a reaction, but we do not die because it's very mild a reaction because we are matching the major antigens. And second, the blood that is received by a patient is always less than the total blood that patient already has in his system. You understand?
Sir, can you repeat what you said?
So it's like diluting just 1 cup or how much blood do we give a patient? Units, in units, units? How much is 1 unit of blood? The bottles that we put, it's 250 cc. You know what is cc? And you
Cubic
can only
centimeter.
allow cubic centimeter. So 250 cubes of 1 centimeter each. Whatever volume you can fill in these 250 cubes is the 250 cc of volume. So that is how we measure the volume of blood. And when you donate, if anyone has donated blood here yet, anyone of the age who has donated blood?
No, sir, but my father has.
Okay, so how much blood did they take from your father?
I'm not sure about the details, sir. I just know he donated blood.
Just 1 unit. Under normal circumstances, they take only 1 unit from every person. In very rare circumstances, if someone is in dire need of that blood and you are the only person with that blood group, then they see that, can you give 2 units? I have not seen anyone taking more than 2 units, because you also need blood in your body, right? And you need time to remake that blood. So blood is remade. So 1 unit of blood donated, your body can remake it within a few days, 2-3 days again. But that's not a problem. Yes.
Can I ask you this question?
Yes, please.
Now, suppose after you donate your old blood and you make new blood, does it purify your liver or in organs which uses the blood?
It keeps your machinery active. Purifies means you have benefits of blood donation, of course. Even if your blood had some toxifications that's growing, when you donate it, you are actually giving out that blood, then you are again putting fresh. It's like It's like aquarium. Anyone has aquarium in their home?
Sir, I had an aquarium.
So how did you used to change the water of the aquarium? You used to take the fish out, keep it somewhere else, then change the whole water?
Yeah.
No, that was not an intelligent design. There's one more thing that aquarium people do. What they do is every third day, take 1/4 of the water out and add 1/4 of the fresh water. That will keep your aquarium healthy forever. You don't need to empty your aquarium ever. Just keep diluting. You understand what I'm trying to say? Over time, toxins will develop, right, in that water. That is your worry, correct? But every alternate day, if you are taking 1/4 of the water and adding some water, it will save you the cost of emptying the whole aquarium every weekend and then taking fish from here to here and then putting them back and all that. You should clean, like, so once in a month or something you can clean, because of algae also develops and all these things. But that is what happens when you donate blood. So it's simple. You have benefits of donating blood. Your system keeps making new and new blood, so it also has to work. But if you donate more than what you should, then your system goes under a pressure, a stress that you don't have blood, so you have to channelize energy into making blood that you are deficient of. But 1 unit of blood, and you can donate 1 unit of blood every 6 months, 4 months actually biologically, but they say 6 months for your safety. If every person keeps donating blood, no, every 6 months or even once in a year on your birthday, if you are donating blood, that's the best thing you are doing actually. And if all of us starts doing that, so I donate blood, I try every 7 months or 6 months. I try to follow 6 months, but then if I cannot follow it, at least once in a year, I make sure that I go and donate a blood. If it's someone I know or someone who's mutual who needs blood. So often these blood groups, blood banks, what they do, you donate your blood and they will keep your blood 1 unit and they will give you 1 unit of whatever blood group you want. You know that? That's called the barter system. So suppose my friend needs B positive, but I am O positive. So I'll go to the blood bank and say, I'll give you O positive 1 unit. Can you give me from the blood bank B positive? Then someone else B positive will come and donate B positive and replenish that pool and take O positive for their friends or whatever, whoever they need. Nice idea, right?
Yes.
So if you can donate blood, please donate blood. It has benefits. So does that answer your question, Hannah?
Yes, sir.
You do not have any side effects. If your weight, your BMI, and your age is okay, you can donate.
Okay.
Yeah. Very good. So what happens in cell-mediated immunity is, coming back to the transplantation thing. Whenever there is an organ transplant, whenever there is a blood transfusion, reaction happens, but it is very mild. And I was just towards the end saying that the volume is very less when we take a blood. So if suppose I need a blood, so I'll take 1 unit or 2 unit at max, which is just 500 cc. But how much blood do I already have in my body? Around 4 liters on an average, maybe. If I'm a big person, very healthy person, tall person, 5 liters of blood can be there. So putting 250 cc in 5 liter or 4 liter already does not make much of a change. So it gets diluted. So even the reaction gets diluted if it is the same blood group. That's why we tolerate it, and it saves life. You are in dire need of blood. If you don't get blood, probably you will die. If you get blood, your body will survive with some mild reactions. So which one will you prefer? Dying or some mild reaction?
Mild reaction.
Of course, right? But in case of tissue transplant, it's different. You must be knowing, if you know anyone who has ever been through a kidney transplant or any other organ transplant, their body never accepts, howsoever perfect the match is, they still have to keep taking some immunosuppressants throughout their life. And why their body cannot accept? Because apart from the blood, the cells also recognize each other or other cells of the body. Okay. So whenever you do a transplant, your body knows, the adjacent tissue, adjacent cell knows that this is a foreign particle, foreign tissue sitting. Now your body cannot differentiate, oh, my kidney was dying, so this kidney is doing a job for me. They just are programmed to kill the foreign pathogen. To an extent, even sperms in a female system are considered as foreign pathogens by the female body. That's why when I was teaching you chapter 3, human reproduction, I was telling you that the vagina is a very, very hostile place for sperm, right? Millions of sperm start the journey, but only few reach the ovum, or sometimes none of them reach the ovum, because everyone gets killed, because the internal reproductive system of the female is built to capture and kill sperms because they are foreign particles. You understand? They are foreign living cells. Is it clear, everyone?
Yes.
Yeah. So what we need to do is, this cell mediated immunity is very helpful under normal circumstances. Any foreign pathogen will come. If it does not go into your blood and antibodies cannot recognize it, then cell mediated immunity will take care of it. But the problems occurs when there is a transplant. So in that case, you have to give immunosuppressants to suppress the functioning of T lymphocytes and this cell mediated immunity. So write down, in case of organ transplants and grafts, skin grafting, people do skin grafting, right? So the best way to do skin grafting is take your own skin or take a synthetic skin that is actually a synthetic material, so body is inert to it. But if you take someone else's skin for a graft, you will have a reaction. So the face transplant, face change, this was a very big sci-fi thing when I was a kid. Like, I was in school, almost every film and television serial, some or the other person is just changing their face with someone. I don't know. Now nowadays we don't see that movies. We are into space and aliens and all these things more. But it was a thing once. But I always used to think how easy it is, like, for me to take the face of someone else. It's not at all easy, because that someone's face is a different tissue and it will cause reaction everywhere. I have to take immunosuppressants all my life to carry someone else's face. So in case of burns, in case of deformities of the due to burning, third degree burn or acid burns, if you have to do a skin grafting, so from the same person, the skin from some other part of the body is taken and grafted to some other part of the body where it is required. So that way your body will not reject it, because it's your own skin. And then you will ask what to do in that skin from where you are taking the skin. So there you can put any synthetic tissue, which will look a little odd, but those will be the softer areas of your body which are not um, exposed. So that can be done. But if you take someone else's tissue, you have to take immunosuppressants throughout the life to suppress this cell mediated immunity so that it does not reject the organ or the graft. Is it clear, everyone?
Yes, sir.
Yes, sir.
So grafting is called xenografting. And just to tell you this thing, interesting thing. So where is science going towards? Science is trying to figure out, is it possible that I can harvest my organs in an animal? So my pancreas are growing in a goat or a pig, for that matter. Pigs are very easy systems to harness. And you must have heard this news. The first kind of a transplantation was done where a person in US received a genetically modified pig's heart. It was genetically modified with human genes. So a pig and a human, basically it's a pig heart, but it worked in the human. And he survived for, I think, around 2 months and then died of the complications of rejection. But 2 months, that's a big thing, right? Anyway, that person was going to die. He just got 2 months of extra life. I don't know, he got a life in hospital only. But if a person can live for 2 months today on a pig heart, 50 years from now, everything is possible, right? Can you see the possibilities? So everyone might carry one pet pig with their organs growing in the pig. So when you do not, like, your organs go bad, you can just get a surgery done. So you have your copies of liver and kidneys growing in animals. I don't know how good it is from animal ethics point of view, but science is amoral, as I have said it. We use it for our moral things, but yeah, it can be done. Not today, but sometime in the near future. But today, if we do that, we have to suppress the immunity. So is this part clear? What is B lymphocyte responsible for? What is T lymphocyte responsible for? There are two types of adaptive immunities, cell mediated and blood mediated or humoral. Blood mediated works through antibodies. Cell mediated works through recognition of the self cell and the non-self cell. Everything is clear. Antibodies, what are they? Proteins, their structure, types. Any doubts? Anyone? Okay, so you can write down the last point in cell mediated immunity. And the body is able to distinguish between self and the non-self tissue. Self and the non-self tissues. And the cell mediated immunity is responsible for organ or graft rejection. Is responsible for the organ or graft rejection. Okay.
Yes.
Now, coming to the point, we know about immunity now, both innate and acquired. Now, the point is, there are two types by which the immunity, like it's formed in the body. One is called active immunity and one is called passive immunity. You must have heard of these terms, right? Active and passive. Have you heard of these terms? I'm sure. Active immunity, passive immunity. And there are two more words, which is called vaccination, active immunization and passive immunization, based on the same concept. So let's first understand what is active and passive immunity. Anyone has an idea what is active and passive immunity? You must have studied the active transport and passive transport, diffusion and facilitated diffusion in your junior classes. I'm very, very sure. Not read of this in chemistry? Active transport, passive transport. Okay.
So we studied last.
Yeah, and forgot. Revise, okay? You have to revise, keep brushing up and keep connecting the dots. Knowledge is not exclusive, anything you study, we for the sake of our learning and for expertise. So I'm an expert in biology, so I'm teaching you biology. Someone who's an expert who has spent more time or in physics, so I've not spent that much time in physics or chemistry or mathematics that I've spent in biology. It's not like I don't know other things, but it's better that I teach you this because I know this the best. So it's better that someone teaches you physics. So these divisions are just for this matter, so that you can study it as different subjects, but it does not mean they are entirely exclusive of each other. Science is all connected, right? In earlier times, they were just philosophers and scientists. They were not physicists, biologists, chemists, computer scientists, data scientists, mathematicians. They were just logical thinkers. So active immunity, as the word says, something is actively happening, right? Something is happening where the body is actively participating in it. So simply, when your antibodies are produced in your own body, that is active immunity. If I'm producing antibodies in my own body towards something, so that kind of immunity for me is active immunity for my body. And when will I produce antibodies in my own body? When do we form antibodies? What causes antibodies to be formed? What triggers antibody formation? People, come on. And know that you know this. Why a baby born or inside the womb will not have any antibodies of its own? Am I not audible, or and no one is speaking? Fatima, are you there?
You're audible.
I'm audible, right? So are you people blank with the question? When do we form? When does our B lymphocytes produce antibodies? Whenever there is an infection, whenever there is a pathogen that enters our body, our B lymphocytes produces antibodies against that pathogen. Okay, is it clear? Antibodies are only produced after an encounter, after an exposure to the pathogen. Is it clear, everyone?
Yes.
Yeah. Okay. Now, if I want to use this concept, if I want to utilize this concept for my own benefit, can I do this? Can I do this? For COVID, and all of you are vaccinated? Let me spend the last 5 minutes on this. All of you are vaccinated or not?
Yes.
Yes.
So when you took the vaccine, did you figure out what is going inside your body, or you just took it because everyone was taking it? There are many people who actually rejected taking vaccines. They were like, we don't want to take the vaccine because we are not sure what's going inside our body. How do you know that they are sending good things in your body which is going to help you? So what was in your vaccine which you took, Atif? What was in your vaccine? What did they put in? What did they put inside you? You don't know? Yes, no, I could not hear you. You unmuted, but you were not audible.
So like as far as I know, like vaccines contain like dead pathogens. So in this case, you can say that they're like putting the dead COVID
So,
virus in our body, so our body can
so
produce antigens.
you were okay with it?
Yes.
You were okay with them putting dead antigens in your body? Dead COVID, a virus which has just produced menace. Everyone is trying to avoid that virus, and then someone comes and says that I will kill the virus and put it in your body, and you just agreed to it. Don't you have skepticism that what if the virus is not completely dead? What if the virus can revive back in my body? What if it can still replicate? Because we know in case of viruses, the virus protein code does not have to be intact. Even if the DNA goes inside, it can make copies of itself, right? Many viruses don't enter in our cell, they just send their DNA inside. So you were just okay. That's very bad, being a student of science. See, that's the problem, with all of us combined. We don't understand what's happening to us. And how did they convinced? How did scientists? How do you think that the community I belong to managed to convince everyone that, see, anyways you are dying, okay? So let me put virus in your body. If you survive, okay. If you die, so okay, no, you're dying anyways. How do you think we managed to survive? How do you think we managed to what do you say? We managed to convince everyone. COVID was a new virus, right? We knew nothing about it. We knew very less about it, about what their symptoms are, about what other things of strains are. When we started making the vaccine, everyone started making the vaccine. So if You're right. So there have been vaccines made, because none of you are speaking, so I think I should not ask more questions. And even someone else also said yes for vaccine. Who was it? Hannah, was it you? So I'll just ask one final last question to you. Are you there, Hannah?
Yes, sir.
Which vaccine did you take?
Sir, Pfizer.
Pfizer. Okay. So Pfizer you take, and you know that what was in your vaccine? Just the same
Yes.
question.
The saying that dead COVID was inside the vaccine. So yeah.
They told you that there was dead COVID virus inside your vaccine.
No, sir.
So where did you get the dead COVID virus from?
I think from
Because
dead people, I guess.
so they waited for the people to die, then they took the viruses from the people, killed those viruses, and put them inside you, so that if it does not work, you also will die.
It sounds true.
No, we scientists take care of things, right? So it's very difficult to convince someone with the technology we have right now. We don't need dead pathogens to make vaccines against them. When polio vaccine was developed, it was developed the same way, like you and Atif are telling, and that's how our knowledge of vaccination is. So we didn't bother to check what vaccines shot we are taking, and we took that. It's even worse than those who denied to take vaccines. So I feel pity about those who denied to take the vaccine, though it's their right. They are within their fundamental rights to deny not taking vaccines. But I feel even bad when those who are taken the vaccines and those who actually belong to the education system, who are students, don't know what they're taking. I cannot expect everyone, even my parents don't know what was in their vaccine, but they took it. But they didn't study science, right? So I cannot blame them. But I know what was in their vaccine plus my vaccine plus all of your vaccine. So there are two ways to make vaccine. One is you neutralize, like you make the pathogen dead, which we call it heat killed pathogen or attenuated pathogen. In case of polio, we did that. And then you send that pathogen in the body when we are sure that this pathogen is not going to cause any harm. This pathogen cannot revive, cannot come back to life, cannot start synthesizing or secreting something toxic. Now, what happens when we put dead pathogens? The concept is same. We are utilizing the fact. Remember, I told you in case of acquired immunity that the first encounter is always what?
Low intensity.
Low intensity and slow, right? And that is where the body meets the pathogen for the first time and tries to make a plan to kill it. Now, what happens is normally in case of COVID, before the body finds a plan, the COVID replicates so overwhelmingly that it takes over the system and starts showing symptoms, and you are severely ill. Then you need hospitalization. Your lungs are affected, infected, filled with water, pneumonia like symptoms appear, and your body cannot do anything on its own. Okay. But if you give a dead pathogen, your body can take sweet time to analyze, think, because that's a dead pathogen. Anyways, that's not going to attack back, but you can keep attacking it until you find the ways to kill it. It's already killed, but finding the ways to kill it is finding the ways to recognize it, and your cells can come and engulf it and digest it. So that's how we make vaccines. But now all of you, the vaccines that you have taken are just the RNA vaccines, in some cases protein, but the RNA for what? COVID is a vaccine, is a virus which is spherical with these spike proteins on the surface. That's why it's called corona. What does it look like? Does it look like the surface of the sun, the way we used to draw sun as kids? Yeah. Inside there is a RNA, the genetic material. If you kill this pathogen, heat kill this pathogen and put this whole pathogen inside, we don't know how the body is going to react to it. So what to identify and to make a response against the pathogen, we just need any one component of the virus to show our body that, see, this is a component you have to recognize and know that whenever this component you see, just send antibodies, make bind multiple antibodies with multiple of these viruses and control their infection. So what we take is, we just take the gene for this spike protein. So this spike is a protein, right? This is a protein coat, inside there's a nucleic acid. So in this RNA, let's say from here to here is a gene that codes for this spike protein. We just take this RNA, put it in a vector. Vector is just like package it as an Amazon packing, and then inject it in your cells. So this RNA goes in your cell, and this RNA is translated by your own machinery. And when this RNA will be translated in your cells, what will be formed in your cells? Just the spike proteins, lot of it. Now this is a foreign particle, yes?
So
And it's enough. You have a foreign particle against which you have to make antibodies. So our system, and can these spike proteins harm you in any way? Can they do anything?
no.
No, because they are not even dead viruses. They are not even viruses. They are just a part of the virus. It's like to train someone, against to train someone to have a defense against a tiger. You are just showing them the tail of the tiger. Is the tail of the tiger going to kill them?
No, sir.
No. So that's the same concept. Now we have the technology to make RNA vaccines. So our vaccines are RNA vaccines. These spike proteins are formed. Our bodies form antibodies against it. Lots of antibodies we make, and then we find that we can actually fight against this. Those antibodies are formed and stored in the body. That's where we get the vaccination effect. We get antibodies in our body. Next time, if the actual virus enters, our body will have the second response, and secondary response is always
High intensity.
high intensity. So it's like a training. You trained your soldiers against a particular enemy before the enemy came, just by showing the photo of the enemy, telling about the characteristics of the enemy. And that how I have to fight this particular enemy, because this enemy has these characteristics. When the enemy comes, you are already prepared. So that's called a false encounter. Vaccination is like generating a false encounter, a pseudo encounter, so that your body can be ready. So that's for active immunity. You have to make antibodies in your body. So for that, you can either send dead pathogen or the RNA for any protein of that pathogen. Passive immunity is when you don't have time and you directly have to send antibodies. We don't have antibodies preformed against COVID. We only have RNA vaccines. Fatima, do you understand? We are not sending antibodies in the vaccines.
Yes, sir.
Yes. What are we sending? We are sending the mRNA for the spike protein of the COVID in our cells. Okay. Cool. Or any other, like not just the spike protein, or any other protein or receptor or component of the pathogen. Okay. Very well. So that's passive immunity, when we directly send the antibodies. This we do in case of, have you seen snake bites? Anti-venom injections are given to people who get snake bites, right? So the venom has already entered the body. Now it's not the time to tell the body to start making antibodies. You have preformed antibodies, inject it and let the body fight. Mothers do it when they transfer antibodies from through placenta and through colostrum into the baby's body. It's a passive immunity. It's being passed on. That's why it's passive. The immunity is itself passed on, but it will not stay there because we didn't form it. We just will use it when all the antibodies are used up. Either we have to form new antibodies as a memory, or we have to do an active immunity. Is it clear, everyone? So you
Yes.
have to read active immunity and passive immunity and vaccination and immunization from the NCERT. And in the next class, we'll go to allergy, autoimmune disorder, and I'll take AIDS as the first disorder that we will study. Okay. Any doubts? Anyone has any doubts? No doubts? Okay, then please go and revise, sit, think about it, think about your vaccines, go on internet, search about which vaccine, Pfizer, AstraZeneca, Covishield, Covaxin, Sputnik, whatever you have taken, read what are they, it's available. Know that what's happening to you, how it's helping you to fight against COVID. Okay, because this is what you are studying. So take care, and I'll see you in the next class.
Thank you, sir.